Welcome everyone. We're gonna start the fourth session of the EANO Youngster Journal Club. I'm Antonio Fuentes-Fayos, a Youngster committee member responsible of the organization of these journal clubs and a postdoctoral scientist at Cedars-Sinai Medical Center in Los Angeles, doing the transition to another institution soon. And who we are, what is this? The EANO Youngster Journal Club is 1 hour webinar discussing cutting edge research, looking for the last publication, following an interdisciplinary research between basic, translational, and clinical, and taking advantage of lively discussion and the networking opportunities for youngsters. Who are we? We are a group of enthusiastic scientists and clinicians. We did a perfect balance between the different fields and different countries, and we work hard to make this journal club possible. So, as a novelty of these sessions from the last one is that we accept youngsters presenters that are out of our working force. So yes, you can contact me or any of the people in the team to participate in the next one, to propose any papers, and be active. So just a reminder that the EANO 2025 is coming, and we're gonna have different sessions, the mentoring sessions, the youngster session, and the networking event. Some details still to be determined, but just check it out, the EANO social media and the website, and it's gonna be updated soon. So we are close to start this journal club. Just another reminder that all the journal clubs are in YouTube, so you can check the last sessions if you want to participate and know how this work. For the lively discussion, just use the question and answer because we are not monitoring the chat. So just put your question and answer. Put your question in that box, and we are gonna transmit to the discussant. And thank you for your participation. And also, after the journal club session, we're gonna send feedback survey. So please fill it up because we take in account that answer to improve the quality of this journal club in the following ones. So today's paper is the prospective multicenter validation of a platform for rapid molecular profiling of central nervous system tumors. And we have the honor to have as a presenter as Dasha Fedorushkova, PhD, MD candidate at the Netherlands Cancer Institute in Amsterdam. And as a discussant, Barbara Kiesel, an oncologist in the Medical University of Vienna, Austria. Areeba Patel, postdoctoral scientist in the German Cancer Research Center in Heidelberg. And Felix, Sahm, professor of neuropathology and head of the division of molecular neuropathology at German Cancer Research Center at Heidelberg, University of Heidelberg. So, Dasha, the floor is yours, or I should say the screen is yours. So go ahead, and don't be shy to ask questions.
Thank you so much, Antonio. I'll just quickly share my screen once you stop sharing yours. Yeah, awesome. Let's see. I think I'll just click here. Perfect. And then I'll switch this. I think you'll be able to see the presentation now full, right? Just as a confirmation?
Yes.
Yes. Okay, awesome. And I'll just get the pointer out and then we can get started. So good afternoon, everyone. Thanks so much for joining us at this journal club. As introduced by Antonio, my name is Dasha Fedorushkova. I'm a PhD candidate from the Netherlands Cancer Institute in Amsterdam. My PhD work focuses on radiosensitization in glioblastoma. But today I'll be talking about a slightly different topic. So we're going to dive deep into this newly published nanopore sequencing workflow for CNS tumors, as well as NMP Flex, a new methylation classifier. And the article that I'm presenting today was published in Nature Medicine last March from the groups of Martin Sil and Felix Sahm, who is present here today, from the DKFZ in Heidelberg. So before diving into the article, I think I'll just briefly give an introduction to the topic. As we all know, methylation status of CNS tumors play a super important role in diagnostics and treatment. And WHO 2021 classification especially underscores the shift towards the evaluation of like a wide range of molecular alterations, including also methylation-based classification. So if you want to make use of this methylation-based classification, you first need to think about ways of analyzing or sequencing, if you want to call it that way, the methylation profile of a patient's tumor. This can be done in several ways. I think most of the methods that I'm naming here are actually already outdated. I think the most common ones are whole genome bisulfite sequencing or array hybridization, as known from the Illumina methylation arrays. It's important to note that if you want to do these procedures, they are quite labor intensive to obtain, which can lead to a long turnaround time in the clinic. Now let's say you analyzing this data and now you want to actually use it for your patient. If you want to make well-validated statements on the methylation data, you need a reliable classifier. There have been, of course, various DNA methylation classifiers published, but I think the most common here and the most well-known is, of course, the Heidelberg classifier, which I think most of the people from the audience will know. So that is just brief introduction to the article that we're discussing. Now, the authors of this paper decided to look into a more, let's say, efficient way of sequencing. This paper consists of two major parts, one focusing on the nanopore sequencing workflow for CNS tumors, the so-called rapid CNS, and the other focuses on a new platform agnostic methylation classifier, the NMP Flex. I'll start this very brief introduction by talking a bit more about the rapid CNS. And to explain the rapid CNS, I'll first dive into the nanopore sequencing. So nanopore sequencing is a DNA sequencing technology that works through passing individual strands of DNA through tiny, tiny protein pores called nanopores, which are embedded in a membrane. As the DNA moves through these pores, each nucleotide, so A, T, C, or G, causes this characteristic change in electrical current, which is running over this membrane, which is then also recorded in real time. And these current patterns are then decoded to determine the DNA sequence, which is passed through this nanopore. Now, unlike traditional methods, nanopore sequencing does not require any DNA amplification or chemical labeling, which then allows for rapid, portable, but also especially long read sequencing. This includes the direct detection of DNA modifications such as methylation and makes it well suited for applications in real-time profiling setting. Now in this paper, a specific feature is applied called adaptive sampling. Adaptive sampling allows the system to selectively enrich for specific DNA regions in real time. As the DNA strand begins to enter the nanopore, the system can quickly analyze the initial sequence and eject unwanted molecules by reversing the voltage, prioritizing sequencing of, for example, target regions as, for example, tumor-relevant areas. This will improve your efficiency even further. Now, the rapid CNS that the authors describe in this paper is essentially a pipeline which is able to run on various nanopore sequencing devices. It includes various parameters, as you can see here on the slide, and it was run and established on fresh frozen samples from two institutes, both archived and prospective samples. I think it's quite impressive to note that in both centers managed to have an average turnaround time of 30 to 40 hours with this rapid CNS workflow compared to an average clinical workup of DNA methylation profile of several weeks. So it's important to keep that in mind when we go through the rest of the results. Now, As a first quick overview, we'll see here the diagnostic samples for both institutes, as you can see here on the sites, and an overview of the concordance for the rapid CNS, with the availability of the NGS and methylation area here on top, followed by the concordance with the acquired rapid CNS data, which you see here below. I won't go into deep right now. I think we might be able to do that with a further Q&A, but just as a quick overview to see what kind of samples there were in the paper. Now, multiple examples are given of glioblastoma, where the rapid CNS predicted well in cases with multiple focal alterations, which are highlighted here in the methylation area, and then here in your rapid CNS corresponding profile. Furthermore, in figure 3D, we can see a Sankey plot comparing the performance of the MMP classifier, so the Heidelberg classifier that most of us know, with the prediction of the rapid CNS based on classes. Each flow that you can see here represents the number of cases transitioning from a category in MMP to a category in rapid CNS, with the width of the flow indicating how many samples were classified in each pathway. Overall, this plot will highlight how well the classifiers agree or differ, perhaps, in classifying the same tumor samples. As I said, it was going to be a rather short introduction for the rapid CNS. We can spend more time in the Q&A on any questions that the audience may have. For now, I want to continue to the second part of the paper, which is MMP Flex. Again, to explain a bit more about MMP Flex, first need to go back to MMPRF. So the MMPRF, or the molecular neuropathology random forest, was a key breakthrough from Heidelberg, which enabled the classification of CNS tumors in these distinct methylation-based subtypes. However, I think as you all know, this particular image, it was tightly coupled to a specific technology, so the Illumina and the EPIC arrays, which are short read based. As molecular diagnostics evolve and labs are moving forward toward their newer technologies such as nanopore sequencing or other methylation platforms with different data structures and so on, the problem arises that MMPRF is not compatible with these platforms. So the authors decided to kind of tackle this challenge by designing MMP Flex. It was designed to be a cross-platform methylation classifier that, well, works across different platforms, arrays, nanopore, GBS, and uses genomic region-level features instead of fixed probe sites. They hope to have it maintain high accuracy while being more adaptable to all these new changes in the molecular diagnostics. And they do that through having a gradient-boosted machine learning model that uses these binarized methylation values. So it's a simplified O1 representation of whether every genomic region is methylated in order to eliminate this kind of technology-specific variability, which would then enable this cross-platform molecular tumor classification. If we have a look at the validation of this MMP Flex, we can see, similar to the Sankey plot I've shown before, in the top figure, a comparison of the predictions performed by MMPRF compared to the MMP Flex. And if we then dive into the accuracy, we can see in figure 4C on the left, the 6 different technologies used, with the left bars showing the subclass level accuracy, aka how well the MMP Flex identifies very specific tumor types. And the right, or the more faded bars, show family level accuracy. So how well does MMP Flex identify the broader tumor groups? What we can see here is that even subclass accuracy drops slightly, family level classification remains robust, which I think is actually quite crucial for a lot of treatment decisions. In the middle of this particular figure, we see a distribution of prediction confidence, so scores from 0 to 1 for each technology. Higher scores indicate more confident and reliable classification, and I think it's important here to note that the rapid CNS had a lower average confidence, probably due to an average missingness of 60.1%, which you can see on the right with the missing CPG sites, which likely affected its accuracy. Lastly, I want to talk to you about the intraoperative reporting, of course. I think the most exciting part of this paper. The authors wanted to test this technology for intraoperative sequencing, for which they first ran some kind of reconstruction of reads generated from these intraoperatively frozen sections. And during this simulation, around 80% of the samples were assigned the correct methylation family within 50 minutes. Of course, this was exciting and called for an actual real-time intraoperative sequencing, which they then did in 18 different samples in Heidelberg and in Nottingham. And figure 5b demonstrates the predictions over 1 hour of sequencing for 2 individual samples run at each center independently. And as I understood from the paper, I think a prospective study to assess the impact on the outcome is being prepared. So maybe we can have a little more information on how that's going. I think I'm already arriving at the conclusions of the paper, which is that we've seen a successful validation of a rapid molecular profiling platform called Rapid CNS, including intraoperative use. This Rapid CNS is powered by adaptive nanopore sequencing, which enables on-demand real-time selection of tumor DNA. And important to notice, it's a workflow which could be impactful in settings lacking high-end molecular pathology labs, which is something interesting moving forward in the time of new molecular diagnostics. I would like to kickstart the discussion with a couple of discussion points, unless there's any other questions in the Q&A which have popped up in the meanwhile. So I thought maybe we could start and talk about the potential impact that you think this paper has on clinical decision-making. What are the costs and feasibility of implementing such a nanopore sequencing workflow in a low middle income setting? What do you expect to have as infrastructural, technical, and personnel requirements? And also maybe we can talk a bit more about the limitations of the MMP Flex, about the cross-platform harmonization, and how to improve this missingness, essentially. So yeah, thanks so much. And maybe we can get started on discussion.
Thanks, Dasha. I think that we can open the discussion. There are not any questions in the Q&A box yet. So again, don't be shy. You can do anonymous questions or put your name on it. And then we can start with your question about what is the potential impact on clinical decision making of this paper, from the paper to the reality. Areeba, Felix.
I think Felix would be best answering this question since he's the clinician.
Yeah, I can do that, of course, if you want. I'm also very grateful always if pathologists and neuropathologists are called clinicians and not something else. So that is nice to hear. Thank you. And I think that is true in a way because, and that is the topic of the question, the diagnosis, of course, somehow determines the clinical management. And the earlier you have the diagnosis, the earlier you can advise the patients on suggestions how to manage in the specific case. And we oftentimes have the situation that we get a multidisciplinary meeting for a patient and we do not have all results yet, even if it is 2 or 3 weeks later. So having this platform here, we here in Heidelberg have typically within the next day or 2 days later or so, we already have results from this platform, which of course is experimental by now. We are currently putting that into the accreditation process, but it's still formally it's experimental. But of course, we have already a very solid indication. And as we see from the data, it turns out to be then also true with the conventional platforms. And therefore, once that is fully accredited, it's of course transformative because you do not have weeks of waiting time until a final integrated diagnosis. That's the first part. And the even more superior part is when you have it intraoperatively, as we have shown in a subset of the cases. So then the surgeons can decide if they go for more aggressive resection, for less aggressive resection, if they know the tumor type, the tumor malignancy, and if they know if maybe there is a target or not. And I think there are different possibilities actually of how it changes treatment. And it could even change treatment if we have something that needs to remain in the cavity, right? which there are currently not so many options for brain tumors. But in theory, if there would be something upcoming, then we would know during surgery already what treatment could be left into the cavity after resection.
And what will be the cost of this in the hospital? Like, where is the investment that the hospital need to do to apply this kind of technology platform? Like infrastructure, personnel involved in this?
Yeah, I think Areeba
Because
set up that in so many institutions. Meanwhile, I think she can explain that very well.
Yeah, thanks.
uh,
Well, it was quite a labor of love, but yeah, so I think it depends on which device you want to set up, because Nanopore has a lot of different devices that they offer. The most accurate of them is, of course, the Promethean, which is a bit more expensive, for example, than the MinION or the GridION, which are these small ones that are as big as your cell phone. I think, and also I think it would depend, right, on different geographies, how much Nanopore sells them for. But I would say about it would be in the range of, I think, 50,000 or something. If you do the most accurate platform, if you get a MinION, which is a much smaller one, it's probably,
Yeah.
I think it costs like 5,000, just the device. And then you have probably a laptop that runs this. So you have a GPU powered laptop, which is again around the same price. So I think you're still seeing this within a 15,000 euro. This is with euro prices, right? So I don't know, maybe
Yeah.
we sell it for different prices, but I would say the initial setup is probably much. And then of course you need to train personnel. But the wet lab workflow is way easier than it is for many other sequencing. So you do it within a few hours instead of the 3-day incubation and stuff that we have with Aries, for example, or NGS. So it's a bit easier to learn as well. And I mean, and in Heidelberg, right, a lot of our master students and HEV students, they also can do this quite easily, even when they start.
Yes.
So it's not something that you need a well-qualified technician to be able to learn it.
Thank you. We have a question. We have a question in the chat. Please use the question and answer box. But Katrina Kurian, what is the difference between the Robin software and MMP Flex?
Yeah, that's a very interesting question. I mean, so the difference is, first of all, that MMP Flex is the classifier, and Robin is a platform that hosts multiple classifiers. And in Robin, we have MMP Flex anyway at the end, where then it does the classification using MMP Flex as well. And Robin, again, is very similar to Rapid CNS. So essentially, we work together with Matt to create both these platforms. So it's a platform versus a classifier, I suppose, is the answer to that question.
All right, thank you. Let's check. More here. No more.
I have to say, when I look into the attendees, that it's very nice to see that we have such an international attendance here. So I really have seen people from South America, across Europe, obviously, and even to the Philippines or so. So that is really great that we have such a wide reach with the EANO webinars here. Really nice.
I mean, normally we have a really good balance, and even people from outside. So our enhancement in social media is working.
So
As
now that we
this-
not have Barbara here, which is unfortunate because she could talk more about the neurosurgical perspective, of course, of these applications. And she could also possibly talk, and it would be something that I would also like to hear from the audience, if you have thoughts on that, on other intraoperative profiling technologies. So there have been very high-ranking publications also on Raman spectroscopy, for example, which also gives you information during surgery about the tissue. And there are, of course, also other technologies that are emerging. So maybe someone has experience with that, comments or questions to the comparison of both, or anything that would most appropriately be represented by Barbara, obviously, but I think there are some technical issues for her attendance.
Yeah, she's still trying to connect. Oh, well, we can continue. In the meantime, that Barbara is trying to join us. I think that Sofia Scioli raised her hand. Please use the question and answer because we cannot open mic yet. We are trying for the following sessions to be more interactive, even showing the camera of each other. But if you can use the question and answer.
maybe in the meanwhile, while Sophia is asking her question in the Q&A chat, we can maybe continue about the intraoperative samples, because I was quite curious what would happen if there's a low tumor purity or stuff like that. How do you account for, what quality control measures do you have that your workflow just keeps running despite having like a low purity of your samples? So what did that look like?
That is actually an important factor, because what we have as a major difference between the conventional sequencing and the nanopore sequencing, at least in the intraoperative setting, is that the sequencing depth is much lower with the nanopore sequencing. So if we use Illumina sequencing for tissue, we have 100 or 1,000-fold coverage or read depth. For liquid biopsy, you have even 10,000-fold or something like that. And here in the intraoperative setting, just because of the time constraints, it's maybe 30, 40, 50x or so much lower, which means if you have a tumor cell content of only 1% or so, and you read 50 different DNA fragments, you may even not capture this information of the single mutant tumor cell, and you may not even be sure if you have one read, if that is an artifact or if that is true. So therefore, I think it is in the current sensitivity, it's more a tool for really representative tissue and bulk tissue, not yet for the tumor front or something like that. But we are working on increasing the sensitivity for these areas, both in terms of copy number, of methylation, and also in SNV and indel calling. But in the current setting, it is indeed more prone to errors of low tumor cell content than the conventional analysis, just very specifically because of the read depth.
Clear, thank you.
And if I can just shortly add to it from the more bioinformatics perspective, because what happens is, we had some samples which were low tumor content in the intraoperative setting. And what happens there usually is that you get such low scores with methylation that anyway we cannot have made any decision. And we all just, the copy number plot also is it's very noisy, or you can say very easily, this is not really the tissue that you're trying to target. So you have these 2 layers of security somehow. So you're not making a wrong decision confidently.
And do you have in mind any limitation about therapy that could alter the classification in the surgery room, like any pre-treatment that you apply to the patient and could affect to the post-classification when you do the sequencing? I don't know if I explain too much myself. I'm not a clinician. Like any treatment, when the patient gets into the hospital and you do the first therapy to the patient.
Well, actually,
have you taken account that? Yeah, go ahead.
That is not a major concern, interestingly. So, and even before the classifier was available, there were these studies just within selected tumor types on methylation, like from Hendrik Witt, from Christian Peitler, but also from other sites, of course, that have seen that during therapy and during progression and so on, the methylation profile is still very stable, generally independent of the platform. And the more confidently the platform recapitulates the methylation, the more clearly you can still identify the tumor, even if it is very undifferentiated due to progression. So that is exactly why sometimes under the microscope, we cannot recognize that belongs to a certain tumor group anymore because of progression, because of dedifferentiation, and because of treatment-induced artifacts. But it's still identifiable by methylation. So that is indeed interesting to see. And therefore, so far at least, we could not identify any limitations on that end. But of course, we are still increasing the data. So that's also important to identify as a limitation. We have hundreds of thousands of methylation data from the array, and we have hundreds on the nanopore side. So it's much of a scale, it's still a lot, but a scale fewer nanopore data. So that certainly makes a difference.
Thank you. Okay, the people was getting alive in the question and answer box. So you said, Pinto, it is possible to integrate indirect measurement, like the convolution in the algorithm or analysis of tumor purity?
Yeah, that's a very good question. I mean, even if it's not limited to nanopore, right, in general for all sort of methylation classification, this is a question that we have been working on. I mean, Felix is working on this quite a lot with meningiomas, with single cell data, etc. For example, I think it would be interesting and it would be possible because there are tools that quantify tumor purity from the arrays. With the sequencing based as well, might be relatively okay to call this, but this is something that would usually happen at the end of a run. So if you're looking at something intraoperative, that would be too soon to be able to quantify a tumor purity already. But yeah, it's a good idea, and it's something that a lot of groups are working. I mean, we are also working on trying to see how if you separate the tumor cells from the rest, would that improve classification? Though I would say maybe in some classes it is the tumor microenvironment as well that influences the class itself, but the immune infiltration, etc. So maybe it's a bit of a two-pronged problem that we need to tackle.
Thank you. So Tanya, another member of our team, how did you address the limitation of cross-platform harmonization when integrating nanopore-based methylation data into a classifier originally trained on array-derived data? And can you comment on the risk of misclassification? Wouldn't it be better to have separate platforms? Why harmonization?
Yeah, again, very good question. So when we started off, we started off with a more nanopore focused classifier, right, with rapid CNS and things like that. But what we found very quickly was also because in the original Kappa paper, we had already showed that you could use whole genome bisulfite, for example, which was probably the only major technology at that point, apart from arrays, that could do methylation, and it worked really well. And at the end of the day, so our thinking for this was, and I think also others who have developed these classifiers would also think the same way, is every site would either be methylated or unmethylated. And if you bring it down to that, with the array, of course, you get much more granular scoring. So you could get from 0 to 1. So it's 0.1 and 0.45 even for the methylation values. But for sequencing, usually you're going to get 0 or 1. So what we did was we took the array data, binarized it. So every site would then be only methylated or unmethylated. And then retrained this, or not retrained, but newly trained a classifier based on this binarized data. And then sort of the hope is, not the hope, it's also something that we've seen, right? Is that this then translates also to other technologies, because if you have to get accurate methylation values from any of these technologies, they have to at least accurately tell you if these sites are methylated or not. So 0 or 1 at least. So that makes it possible to have these classifiers which are platform agnostic when you bring it down to this binarized sort of setting.
Cool. So next one from Jan Werner, a youngster former. Could you combine it with fast glioma to reduce sampling errors? It was a recently published methodology on Nature referring to that.
Yeah, that's a very smart question. Thank you, Jan. Yeah, indeed. Actually, that would be a nice combination. And just 2 days ago on a neurosurgery meeting, we were also talking about this possibility of the complementarity of these technologies. You could use the fast glioma to see what is actually very suitable tissue for the rapid CNS, because it can identify tissues where there is a high tumor cell content and which are really representative. And then you have done rapid CNS interoperatively, and you know what type of tumor it really is, astrocytoma, IDH mutant, oligodendroglioma, glioblastoma, or whatever. Or you may even see it's not a glioma by the rapid CNS analysis. And then based on this certain knowledge, you can then use fast glioma to go doing the procedure. The surgeon has progressed in the resection, comes to the margin of the tumor, and then can use the fast glioma detection to see where there is actually the infiltration zone and to what extent there is infiltration zone. And interpreting this infiltration zone fast glioma signal is, of course, much safer if you know if it is actually a glioma and what type of glioma it is. So it's actually very complementary, and I think a very nice combination of technologies to be safe on what you are working and doing surgery on, and then to explore the margins of what you are doing surgery on.
Oh, we have another question. This one from Anonymous. How is the methods performance of classifying MGMT promoter methylation compared to other commonly used methods? I think this is an important aspect from a clinical perspective.
Do you want to take that, Felix? I mean, I can say what the accuracy is.
Yes. please.
Yeah.
So the accuracy was about 91%, concordance between the array and NAT4, and I think that's higher than what is also currently between phylosequencing and array, probably, if I'm not completely wrong, Felix.
Yeah, so I would not have known the number. I would have need to look it up. Thank you, Areeba. Excellent. But what we also considered when looking into specifically that number and the precision of MGMT promoter methylation assessment, we also noticed that there is no real study that defines what is the gold standard of MGMT analysis. Most, but by far not all, do pyrosequencing. Some do methylation-specific PCR. Some do more recently dPCR because there are also now, at least for Europe, relevant IVDR compatible tests upcoming. But there is no really large study with glioblastoma that were treated with temozolomide, where there was tested if the predictive value of one or other test would be better or more precise in the predictive value. Since we know methylation array, pyrosequencing, methylation-specific PCR, and nanopore, they are not 100% concordant. They have about a 10% variation in each direction and not overlapping between the different technologies. And therefore, there is actually no real ground truth for these discrepant cases unless we would have a study in which we could see if these discrepant cases would actually behave more according to the one, the other, or the third test. So we can hardly actually tell what is better. And at least the biological hypothesis would be that reading the entire promoter region by long-read sequencing as nanopore is probably more informative in the end than just picking 2 or 4 CpGs, as some other tests do. But we still have not the clinical trial data to really confirm that.
Okay, thank you. Anne-Sophie Vamelis, she has 2 questions. So the first one is, I have read that if the nanopore sequencing rather gives no classification than the wrong classification, if it is unable to come up with the accurate class, is it the similar for MNP flex?
And that's a very good question. And that also applies to not just nanopore, right? It applies to all methylation classifiers. Because also it should be like that if it cannot classify it, unless that's a good calibration. And if Martin Zill was here, he would give you a lecture on good calibration for a very long time. But yeah, essentially that's how MNP flex works as well. That if it cannot come up with an answer, it would rather give you no classification. So it would give you a very low score, which is also a sort of safety net, because you do not want your classifier making more predictions with high confidence, which are not correct. Because at least I would rather, and I think also Felix would agree, we would rather have a sort of, have some cases which are not classified than have any of that are wrongly classified with a high confidence. So yeah, absolutely true for MNP flex and all the classifiers that we try to develop.
So Anne-Sophie has another question, like, do you have experience with reducing the flow cells on the nanopore? Does this decrease the accuracy?
Yes, for sure. So we did this for all of them, for the grid ion, so the min-ion flow cells and the Promethean ones. It does not decrease accuracy, but what happens after the first flush is usually that the pores have become less active. So you have to then sequence for longer. So what the data you could get in 24 hours, now you would get in probably 40 hours. So it's just, the accuracy is not impaired, but yeah, you just have to sequence it for longer than to get equal amounts of data.
Thank you. Josep Pinto again. How do you address intratumoral heterogeneity in methylation-based classification?
Yeah, that's of course a relevant question. And I would say there are 2 dimensions of intratumoral heterogeneity. The one dimension is heterogeneous in terms of more tumor cells or more tumor microenvironment cells. So there are areas where there is more vascularization. There are areas where there are more lymphocytes, microglia, macrophages, and so on. And then there's the heterogeneity in terms of tumor intrinsic heterogeneity, different tumor clones or tumor cell clones. If the heterogeneity pertains to more immune cells in one area, then of course we have less signal of the tumor, and therefore the classification accuracy is lower because we just get the DNA from the immune cells, and that is not so informative, at least not solely informative about the tumor classes. If it comes to the tumor cell clones, then it actually is not such an issue, because since the current understanding is that methylation-based classification is just a readout of the imprint of cell of origin, that does not change. I mean, the cell of origin is at the beginning, and whatever the differentiation the clones show, it's still the same cell of origin in the beginning. And therefore, we see that that is very stable and also very homogeneous across different clones within the same tumor. There can be small variations. For example, in a glioblastoma, there can be areas that have the RTK1 subtype within the methylation classes, and the other one has the RTK2, more or less side by side. And then sometimes we see that under progression, it becomes then a mesenchymal glioblastoma methylation class, but it still becomes a glioblastoma prediction and not an astrocytoma IDH mutant or so, obviously. And also doing progression from subependymoma to an ependymoma, that may also change then from methylation, but it's still in this ependymoma space. So generally, the heterogeneity among the tumor cells is not a concern. It actually seems to capture the relevant heterogeneity in terms of class progression quite accurately, and is not somehow affected by clones that may be more differentiated than the others.
Oh, thank you. I have another question that we discussed in our team. It's like, do you see potential of the rapid nanopore-based diagnostic beyond neuro-oncology?
I didn't get that question. Sorry, what was the exact question?
Like, if you see the application of the nanopore-based technology in the diagnostic beyond the neuro-oncology, like
sorry.
in
Yeah.
other cancers.
Yeah. Yes. So the answer is shortly yes, because every classifier for methylation can also be used on nanopores. So there are also sarcoma classifiers already. There are other classifiers from different groups, not only ours, that are about to be published or are already published. And the technology, and I think Areeba can talk more about that from exploring that already, but the technology is, of course, completely transferable to other tumor types.
So what tissue related limitation could have that, or doesn't have any limitation relating to the tissue that you use for the nanopore? Or maybe in one part of the workflow, if you have any limitation, if you use a different tissue, like going through the nanopore.
I think the main limitation in terms of tissue is that it has to be fresh or frozen. So it cannot be FFPE. So as long as you have that tissue, I think you're good to go. And then for any sort of adaptive sampling based platform, you then need a list of targets, which usually you would have. And then that's pretty much, I think there are also a lot of studies that came out recently, at least in the past 2 or 3 years, which have showed that people have used adaptive sampling for different cancer types, but especially for structural variants. There was a big study somewhere, I think from Australia, I can't remember. But they showed this, that they could look at large structural variants that they would have missed even in short read whole genome sequencing, just by targeting the regions that they knew would be problematic.
Thank you. Then, like, we have Barbara Kiesel finally. Hi, Barbara. Could you hear us? Sorry, we are having problems like to get Barbara on live. We have it. Hi, Barbara.
Hello.
Finally.
I'm
She was dealing.
I think the question, how does the information from rapid CNS change the neurosurgical perspective or the neurosurgical procedure? That would be a question for you.
Sorry, can you repeat it?
Yeah, so we discussed when you get this information from nanopore sequencing during surgery, what does that mean? Does that change your procedure, or aren't you much interested in it?
So in fact,
No.
That was short. Okay.
Okay. We're gonna continue for the next
Yeah,
one.
please
Go
go ahead,
ahead, Dasha.
Antonio.
Go ahead.
So I can
Go ahead.
maybe,
We can save it
Well,
next one.
I have one question because I could imagine that especially this pipeline or this classifier is particularly interesting for rare pediatric, atypical CNS tumors, what so on. Those are, but also, they are also because they are the diagnostically most challenging. So you want the extra help, whatever you can get, because maybe regular pathology, H&E staining, whatever, it doesn't work out. So those subclasses are, I think, by nature also underrepresented in your training data. How do you handle that? How do you hope to improve that in the future?
Yeah, so about the training, right? When we do the training for these for this reference data set, we basically tone it down to the minimal class, and then we train on that. So essentially, of course, there's a major class imbalance, and there will always be just because of the incidence rates, right? But that's something that's already accounted for when we do training. But of course,
Okay.
I mean, we discover new classes. That's again a different thing. That's something that we have been doing with the MNP classifier for many years. Felix and everyone else. That is again a different thing because then you're discovering new classes. But once you have them, when you're training the classifier in the reference set, we take it down to the minimum sample coverage that we have.
Okay, so there's no like, I don't know if I'm saying you're using the right term here, but some kind of overfitting based on like the low samples that you have.
No, I mean, also then with the calibration that solves that problem, right? So then the scores are adjusted according to that.
Thanks.
Right. So, Barbara,
Yeah, I hope it's.
we have you back.
I'm sorry, because I managed it now on the phone and someone was called permanently. And yeah, so sorry. So for the information you get during surgery, I think it's a great opportunity to get this information because I don't know how it's in other departments, but when we do intraoperative diagnostic from the frozen sections, the pathologist usually is not, he can sometimes not really distinguish all the tumors. And when you have more information like the methylation data, and you really know which type of tumor it is, you can really improve your surgical goal. Like, for example, more differentiate between medulloblastoma, ependymoma, that you really try to get a gross total resection, or also in glioblastoma, when, for example, you have an unmethylated tumor, MGMT unmethylated tumor, or a methylated tumor that you maybe try to remove more for the unmethylated tumor, because the postoperative treatment is not working as good as in the methylated ones. So I think for the neurosurgeon would be a great benefit to have all this more information. Did you hear me? Sorry.
Yeah.
Yes, thank you.
Yeah.
Thank you. So let's go for the next one. From your experience in Heidelberg, how many tumor samples can you run at the same time? What is the limit in terms of on data processing?
Good question. That again depends on the device that you have. If you have this GridION, it has 5 flow cells, you could do 5 samples at one time. If you have a Promethean, or the P2 or P2 solo, then you can do 2 flow cells at once. There are a lot of efforts to barcode these samples and run them. They work for sure, but I think if one is looking for something like intraoperative or rapid diagnostics sort of setting, I think 1 sample at a time is better, because then you're getting a lot of information about that same sample at one time. But if one is okay with, a little longer runs, then you can also barcode 2 or 3 samples on the same flow cell and run that together. It also makes it a bit, I think, cheaper to run it.
But I think if we're talking about rapid CNS, the barcoding, of course, heavily affects the adaptive sampling, because you have for everything to also call the barcode, right?
Yeah.
That is then, of course, somewhat on that end less efficient as a trade-off for the efficiency of using a flow cell for more than 1 sample.
Yeah.
So this was the last one.
Can I ask a question? I know I have
Sure.
missed a lot. I'm so sorry. I wanted to know if I read it right. If the nanopore is not able, or your platform is not able to use it in metastases. Am I right?
No, it can. It's just that if, as long as the class is represented in the methylation classifier, one could use it. But a lot of the metastasis classes are not represented. I mean, some of them are, of course, but not all of them. So then the methylation cannot be done. But on the other hand, you still call the mutations, fusions, CNVs, etc. that can be used to make a diagnosis. It's just that the methylation part is a bit lacking, just because we don't have these classes in the reference set.
Okay.
Oh. Okay, we are 8 minutes, so we can talk more a bit. Oh, well. Okay, what is like, do you think that is going towards the new classification of the tumor in the future? Like, this is feasible? This only gonna be like in some hospitals? What do you think, like personally, your point of view? Because the molecular classification of the WHO 2021 is still like being applied in some
That's a good
hospitals.
point, and I think the 8 remaining minutes will not suffice to discuss this in detail because that is a really large ongoing discussion, but a good point indeed. And I think also something that Dasha had on her slide for discussion, right? On the one hand, the possibility to buy a comparatively cheap device, I mean, that is like a fraction, a small fraction of the cost of another sequencer or scanner. And you get both. You get the methylation and the sequence information if you even purchase the smallest device from the nanopore things. And that enables then labs that before had no access to molecular testing to suddenly have everything from just a single 5,000 euro or dollar investment, which is still unaffordable for many sites in the world, but at least increases the reach extremely compared to before. So therefore, I think there is extreme potential in technologies like this, and there are also others. I mean, MNP Flex, as Dasha presented, is platform agnostic. So if you, for example, have sequencing capabilities already on site, and so far did not do methylation because you did not want to purchase a scanner and all of the periphery devices, then you now can use the sequencing device that you already have with a bisulfite sequencing approach. So that, of course, also, again, widens the institutions that can instantly use this. More generally, the discussion of the 2021 classification, if that is still a World Health Organization classification, because it mandates so many testings that are not worldwide available, that is indeed very debatable. But on the other hand, the question is also, if you know that certain markers make much of a difference, should you then not enter them into the classification, right? There's always offered by the classification the possibility of saying, okay, something has not been tested. That is very clearly lined out how you proceed then in that case. But on the other hand, it would also be probably inappropriate to stay silent and just say, well, IDH doesn't matter also, because it obviously does. And it should also, to the extent that is possible, motivate all of us to work towards making that available everywhere, instead of somehow being shy of introducing important scientific advances into classifications.
Barbara, you have any point of view of this?
Just for the future, I think it's also good. So it maybe improve also the application of intraoperative treatments, or for example, when you after the resection can put some medication in the resection cavity, and if you have all this information within or a lot of the information within 30 minutes and doing surgery, maybe also this area of treatment may improve in the future. And
Great.
the costs, as you mentioned, Felix, I don't know how it's in the other countries, but for example, in Austria, the government is like struggling with the finances and try to reduce costs also in the medical field. So maybe we will also more use this technique more in the routine practice than all the fancy and costly other devices, and use them only for specific questions, and introduce the nanopore and platforms more in the routine clinic to reduce the costs for the patient or for the insurance in this.
Yeah, that is also what I expect, exactly as you say, and I would also expect that very simple technologies can be leveraged much more with new approaches to these simple technologies than before. I mean, we know from many studies from Adelheid Wörer, from colleagues in the US, and from a project that we have running here, that we can actually read out a lot of information from an H&E if we use artificial intelligence that is trained to identify tumor types, to identify copy number alterations, and so on. So that would then, exactly as you just said, Barbara, that would help us to triage cases where it is obviously clear from the H&E and the artificial intelligence that it is tumor type A, B, C, or whatever. And on the other hand, identify cases where we need the additional testing, which is then more expensive. But I think advancing these very simple H&E-based tests that are surprisingly powerful, almost can't believe that in some instances, that is also a very promising way of making that much more widely accessible.
Great. We have another question in 2 minutes to finish. So if you can answer briefly, how will molecular pathology and AI change the work of neuropathologists in the future, farther away from microscope and histology? If you can answer that in 1 minute.
Would be funny if someone else would answer that now instead of me, but if, maybe that's a question.
Focus on you all the questions.
Well, I think before a pilot starts to use an autopilot on a Boeing 747, he or she trains on a very simple plane in the desert of Texas or Arizona or so. Therefore, I think it always is needed to train the basics so that you can then control what the technology is doing. And yes, in daily work, it makes it much more efficient. It also is a safety net in a way, but it is somehow reciprocal safety net. You still need to be taught to also identify what is actually going on with the technology and to make sure that that is correct and that is accurately used.
Oh, so right on the time, Felix. Thank you. So we're finishing. Thanks for joining, and sorry, Barbara, for all some issues.
I don't know. I'm so sorry.
Yeah, it was so stressful for Areeba, me, and you, everything. So that's it. See you in the next session. Also, thanks for joining, Dasha for presenting. And please, all the attendants, fill it up the feedback survey so we can improve this a little bit and we are trying to improve the live discussion in the future so we can reply answers. So see ya.
Thank you. Bye.
Have a nice one. Bye.
Thank you.